So we have chapter 2, cellular organelles and cell membranes. So we have the marker enzymes and the nucleus, and so we have the DNA replication, RNA synthesis, and RNA processing and ribosome synthesis. We have the endoplasmic reticulum, and we are having detoxification of various drugs, and so then we have this Golgi apparatus and things, and the glycoproteins, protein sorting, packaging, secretion, lysosomes, and the clinical applications, and all of that. And we have post-mortem autolysis, and we have inclusion cell, eye cell disease, mannose 6 phosphate, and peroxisomes, hydroperoxide formation, the catalase and peroxidase, and the adrenoleukodystrophy, and Zellweger syndrome. And that is, the proteins are not transported into the peroxisomes. This leads to formation of peroxisome, and the peroxisomal goes inside the cells. So we have primary hyperoxaluria, and defective peroxisomal metabolism of glyoxylate derivative of glycine, and all of the things. We have the powerhouse cell and electron transport chain, and cytochrome P450, and steroidogenesis, and then we have the plasma membranes, and nucleotide phosphatase and alkaline phosphatase, so the ectoenzymes, and we have the fluid mosaic model, and choline and ethanolamine and serine, and we have the cis fatty acid, and membrane proteins, and transport mechanisms, simple diffusion, facilitated diffusion, and the ping pong states, and then we have a water channel or aquaporins, and channelopathies, and so I remember one madam, usually in biochemistry, she used to stress and say the words and all of that. But certainly, like only anatomy we were able to learn and understand. Physiology and biochemistry. Physiology was okay, and biochemistry also we were able to understand, but through book only, not like very sleepy, and like it was interesting only. It was interesting, but we could not understand what they teach, and like so then this channelopathies, and cystic fibrosis, and Liddle syndrome, and periodic paralysis, and all of this. Just because after schooling, and immediately 1 year we had this engineering, and these words are completely different. These somewhat belong to the Australian Smithsonian University, I think. and these words are different, and so there is abnormalities in proteins forming the ion pores or channels. um, So why would you must have taken us to some site and shown through depictions, but if you're presenting them now after studies is a mere waste, and so but we could have a certain remembrance, but at the time of studies you must have shown some models and all of the things. And amylogenin, and so calcium hydroxyapatite crystals, and the voltage gated channels, and the point mutation in sodium channels, and to myotonia, and so valinomycin and gramicidin, mobile ion carriers, and the channel formers. So then we have alpha 2, beta 2, the cardiotonic drugs like ticoxin, Cubane, like Aubane, and then we have fluorhizin, an inhibitor of sodium dependent co-transport of glucose, especially in the proximal convoluted tubules of kidney, produces renal damage and results in renal glycosuria. Then we have this Hartnup disease, transport mechanism for amino acids, defective in intestine and renal tubules. And we have cystinuria, renal reabsorption of cystine, and we have the phosphate decrease in vitamin D resistant rickets. And so Vasudevan usually used to present me. So my name is Sri Kumari here. So previously my name was Indu Kurana. So Kurana is my grandfather, and here my name is Sri Kumari, and he has presented the book to me. So Vasudevan is Lord Mountbatten of Germany, and usually his origin is Russia, and I'm Queen Elizabeth of England. So he has presented me this book. I think not only this book, all of the pathological books and most of the books. Like presenting means he not give this book for free. He has written it as a whole, and with all of our memories in school, I think. But along with the words and names, and and we have this proline amino acid, and so there were different types of names, and glycine, alanine, valine, leucine, isoleucine, serine, cysteine, methionine, asparagine, glutamine, aspartic acid, glutamic acid, lysine, arginine, phenylalanine, tyrosine, tryptophan, histidine, proline. This we need to memorize. I memorized all of these names, and so very nice, isn't it? We remember the names and remain safe in Romania. This because and the lake was. It was not a lake. The quarry was like getting us into the water, and I don't know swimming also. And how could I remain without getting into? Um, but life saving strategies. But people are there around. But we must remain um, heroic to our animals, isn't it? And must not lose the time lapse, the minutes behind uh, our soils and our spacecraft. So so we have all of that, and we have the molecular structures also. Yeah, nice molecular structures. So how did I miss it? Like if we have written a paragraph and drawn a molecular structure and written it, then we would have had an extra of 1 or 2 marks, which would have made us have a gold medal. Uh, but Vasudevan, really, I was not interested about all of these, just because you made me very deep and sad into my 12th results. Actually, it might be a higher heights. 93% would be a highest of all of the persons there. But I was quite, I don't know, I was restless, and I was not very much interested going into college. I thought I would remain back, maybe continue my arts college, maybe do the work of a chief minister, and maybe I would have had those dreams. Um, but suddenly you made me into the army and lead the army, but I'm very fragile and delicate, so I was not able to make up to it. And we have the classification based on nutritional requirements and all of that, histidine and arginine, and I think these are classification of oil in the kitchen, and for the chicken, and the properties of amino acids, sodium glutamate and aspartamate, mm, ampolites, zwitterions, just some beyond 20 kilometers. It is entirely the field, and entirely Mountbatten is very much responsible, and wherever we go we need to take our Saturn space station, and we need to remember that. And so there is no net charge, and all of the equations he has given. So it was like I have closed my mind for all of the equations. So we finished with the benzene groups in the chemistry. So, and I had not opened my mind for this. And we have the glutamic acids, and we have all of that. General reactions of amino acids due to carboxyl groups, and we have the so where did you get this papyrus? It is just breaking into, always like breaking into, and I hate tearing books. And So this gabapentin, cycloserine, azathioserine, and all of that, and peptide bond formation, and this protein structure and function. Then we have the characteristics of peptide bond and all of that. So the spatial orientations, so G and Ramachandran, and the numbering of amino acids in proteins and all of that, branched and circular proteins. Very nice, isn't it? If you had said it is an arts college, then I would have certainly I would have left memorizing. I would have got through the entire book, but I have read also, but not in a mind to read, so that I had clashes, and so C peptides and all of that. But to an extent, I know all of that about the core of the studies, and epsilon amylo groups, and so and the beta pleated sheet and the collagen helix structure and the tertiary structure and the quaternary structure, and end group analysis, primary structure sequencing. So reading this, I remember about the Sangakiri lorry station. So there's a way from not the Edapadi Sangiri road, but before that there is a lorry station. So that is a train port, I think. So there we see all of these structures and names and all of that presentations. A very nice one. And we have this partial hydrolysis and advanced degradation technique and fingerprinting method, engrams technique, and I just went through all of these, but then we have this salting out, precipitation reaction of proteins, and then the classification of proteins. So like all of these audios, if I went through all of these series, then my entire books collection is closed, and I have got through all of my books, and we have this casein of milk, vitelline of egg yolk, and Glue his pro gamma glutamyl cysteine glycine erythrocyte membrane integrity, and we have this gramicidin S, Bacillus brevis diphenylalanine. We have the Geldal's procedure and the Burette method and the Lowry's method and the spectrophotometric estimation. So beautiful chemistry. I love chemistry, and especially these physical and chemistry names are at a sequence of my mindset, not even biology. So usually I would have taken my masters in physics and chemistry. And we have the classification of enzymes and all of that. People don't think that I am presenting my paper now. I am just having a review and just depicting my experience with learning and how I enjoyed my storybook of sciences. So this author has a very nice mindset, and he has when everyone of other teachers had thought about the um, health of the people, the children, health of the children, and taking care of them and all of that, the food, and so like certain people, certain teachers, they have thought about the food the people, the children's eat. They brought in all of the food products and good cultivation and good health to them, and certain of the people thought about the dresses and the forms and their communication and literature skills, the studies and all of that, and they have taken care of the nice beautiful dresses and the shape, and how do they get connected with others, how do they approach others, and how do they write their own style, modified style, and all of the each unique styles, and certain teachers think about that. But this author who has written has thought about the young old things which everyone would miss, the stories told by the mom, grandmothers, and the family members when they talk, when we listen in the young children, as a young children. And what we talk with our friends, the first word, and the stories, and which we hear to sleep, which we hear to make ourselves calm. He has thought about all of that and made a nice presentation. And usually I like to listen to stories usually, um, and I had this problem of reading, I could read and memorize, but I could not read it entirely, for a longer time. I go hypoglycemic, so but now it all had uh, left. I have started reading a lot, um, as usual, and so then we have all of these and the coenzymes. So we have the nicotinamide adenine dinucleotide, NAD, and we have the second group of coenzymes, adenosine phosphate, and metalloenzymes, the cofactors, and lowering of activation energy, the mode of action of enzymes and serantinases, and action of toxins, and we have Michaelis-Menten theory, and we have this Fisher's template theory, and we have Koshland induced fit theory. So the enzyme substrate complex theory, and the three-dimensional structures of active site of enzyme is complementary to the substrate. So and substrate fits on the enzyme similar to a lock and key. A lock can be opened by its own key only. So then we have a picture depictions and all of that, and we have these exergonic, isothermic, endergonic, and enzyme kinetics, and Then we have enzyme concentrations, substrate concentrations, and the endpoint method, and then the Michaelis constant, and the definition of Km, and the glucokinase, hexokinase, double reciprocal plot, the Line-Weaver-Burk plot, and double reciprocal plot. And so I became allergic to all of these words, because if I start reading, I'll not leave this. And we are having this enzyme inhibition, competitive inhibition, and so clinical significance of sulfonamides, methotrexate, dicomarol, and isonicotinic acid hydroside. Then we have this non-competitive inhibitors. And we have the suicide inhibitors, and we have allosteric regulation, and so we have different varieties of names: feedback inhibition, and tryptophan pyrrolase, ALA synthetase, and glucokinase. I memorized all of these, and with a certain hint. And we have this repression and covalent modifications. And I just read all of these for the one word, and we have the Catal's expression of enzyme activity, and so each of the specialists, so what you must have done, Vasudevan, is that you must have like lack of communication again. You must have asked. So we are about having 16 ways to the different countries. At least you must have taken some 10 people together. So at a time you can address about some 10 people at a time surrounding you, all of them listening, so that like one person you must have given anatomy, one person to physiology, one person to biochemistry, and make a thorough handling of everything along with other subjects. You must advise them to have notes of everything, and mark the changes, and review people about the studies, so that we don't go missed with everything. So this must continue for ages, so we can't leave it as it is. But each one would have written separate different answers for every questions. You can have a combination of that, so you have 100 different combinations of questions answers, and you could easily get into the book. But to the core of it, you must have asked at least some one or two persons, put one as a leader for biochemistry, and have a team of 10 members among the 100, and have a biochemistrical team, so that learning goes easier and very interesting to everyone. So but we did not do this. It was actually just because of the mm, like lack of idea. And okay, we could do it later also. Like even now if you ask them, they will do it, uh, because they know everything about and and we also have the previous batch of us, so that I'm not mentioning about ourselves in the previous batch. I'm mentioning about our elders in the previous batch. We have one more batch who have small kids, small kids, and those batch, they would have taken up our work, so that Vasudevan didn't mention it for us. So we have some extra time for other activities and our own imaginations. Okay, next we have chapter 6, isoenzymes and clinical enzymology. So it's like we having allozymes and polymorphism. And we have so at the first time when I read all of these, I memorized and wrote on the book. But now looking on to the countries and small this uh, um, sets, cinematic sets, and small things, and the plants they grow, and how they look after these people. The people who studied with us in the college only, they have created the practical part of it. Practical means it is not the practicals we do in the college, that is different. This is applied part of the things. They have done a beautiful job, and congratulations. So that just because you have created all of these, this architectural group people, they have helped for these, and also the elders and all of them who had done through this applied practicals, applied things on the land, and the engineers. The previous batch, I could say. So they have done a very nice job, and very appreciable. And and I also request my friends, if you suspect any of these small beautiful works, take a photograph, and or at least take a note of it, and take a short video, and just make it publicized through the media, so that everyone knows about it. And so next like and also the real value of these books lies in that natural shapes and making. So very cinematic also. And and we have the isoenzymes of LDH. We have H4, H3M, H2M2, M3H, M4, and we have H4 forms in the heart, and this flipped pattern. So so we have this muscular dystrophies, and all of this, and cardiac troponins, and we're having aspartate aminotransferase ASTs, and brain natriuretic peptide and markers for cardiac diseases, and alanine aminotransferase alkaline phosphatase. Just because I'm reading this, don't think I'm a first year anatomy student and start ragging me, because I also feel the same. I'm just feeling like a first year student reading. What I have done priorly, I think previously, before this, also before my schooling, also I have done all of these. A certain of these, I think. And nucleotide phosphatase, yeah, I remember reading these chemistries, and gamma glutamyl transferase, and so the primary classes, I think the same is repeated, just because in the higher secondary, in the matriculation syllabus, that is equal to a college status, and after that, this colleges, we will start again with the basic, so that we everyone comes together, and so this is that part which has got caught, and so we have this tartaric acid and tumor marker. So this gourmet side, this all of this industries and the colleges, and the author has combined how much ever he wants to add, and he is feeling very much pretty on writing. So why is he doing like this is that, so we other authors among him, like my dad, my grandfather, my uncle, and my co-colleagues, my friends, all of them, what we do is that we learn and write by our own, and we talk by our own. We just don't have a piece of paper, memorize, and then write or speak. We just speak from our knowledge. But this author is feeling very much guilty that he usually writes in papers and then produce it, and he could not write by his own. So because there is a barrier, usually every of the persons have the barrier, but the barrier is less found in certain of the people of England. So this author is from France, so he has thought that he is less brilliant than all of us, and so to his guilt he has written all of this, but to equalize our writing, he is just he is someone like a mother who thinks to equalize a baby's sound. If a baby says mama, the mother also says mama. So to get along with the baby, so to get along with the friends, this author is appreciating the memory of the people with him, so, and also the memory of the rulers, and so this colon apnea, so there is a nightmare of anesthetist he has written. So see, I have just told about the author, and just the word comes back, and And what else? So succinyl choline is known as choline, nice word. And I am just there was this buscopan medicine given in the hospital. I just forgot about it. We don't use that in Vinayaga missions, but only rarely they use. In medicine, people they usually write the medicines, so I did not know about it. And something like hyacinth, yeah, and I also think insulin is from the plant, and so I think this after this, just because I very well did not know about this biochemistry. Next, the pharmacology was hanging up, so I did not get through. The second time also, I just struggled into the notes. I took the book the first time, and second time the Dripati book, and second time I just um, took the notes, entirely mugged up the notes, and went to the exam hall. And practicals did not answer one or two questions, but anyway, pass. But still, these remain burning in my heart. Because I got some 183 in chemistry, but at the same time, the second volume of chemistry, I was not able to learn much. I don't know why I'm getting these type of diseases due to aging, I think, because we have, and also lack of time. So if you have one day for a class in the college, the next day you must allow the person to be at home, so such that we can finish it entirely. So we can't get past these storybooks as happening, but not everyone knows everything. And the methemoglobinemia and amylases, acute pancreatitis, amylase in urine, acute tumor markers, and all of that, streptokinase, urokinase, pepsin, trypsin, asparaginase, and all of that. And I still remember reading these at the peak hour of my exams. And then we have chemistry of carbohydrates. I don't think I'm talking too much now. Previously, I used to talk to myself, and just I'm sharing my words with others. And so we have this chapter 7. So the DNL isomerase of glucose and all of that, and diastereoisomers, and epimerism of aldoses, anomerism of sugars, mutarotation, anomeric carbon atoms, and 3 representation of glucose structures. So how much do you think a friend would work for me? So he has done his best, and he has given his best, and he had made me into a nice friend and a nice presenter, and all of my friends and families. You can't expect more of that, because a human human eligibility is, that's all, and more less than that. And we have this aldose ketose isomers and all of that, and a reaction of monosaccharides and enediol formation, and the Benedict's reaction, and glucosuria. And these Benedicts and all, I am very much concerned about the titer. So just I could just make a drop into half and quarter and enter into the test tube. So I had that much skills when I entered Benedict's and ozone formation, and still now I could make it, I think. Just because of this, my Chandrayaan-2 mission was running during my first year. It was then second year. So during my third year, I had this INSAT satellite. So I need to run up with it. And all of the happenings of the INSAT, just because we not have a revision of this book, all of the happenings of INSAT was going through as a media information. So it is only for a premium member who had paid for the college. And so the questioners must have not been a premium member, so that's why I am getting all of these questions. And so we have oxidation of sugars and aldonic acid. And anyway, thank you questioners. I remember my schooling and furfural derivatives and Molesh test. And so then we have the glycosides, say glycones, and the sorbitol, malcitol, mannitol, and formation of esters and phosphorylations. And we have the fusion staining, and xylulose, ribulose. Actually, this chemistry is a kind of testing, and if you see psychologically, it is a disease. So certain people have the disease of testing each and every of the living. Like if they have sugar in the kitchen, they used to cook it several times with several dishes and try the taste of it. If they have pineapples or apples in the fridge, they used to cut it and they used to put it in a mixie and have a pulp, or used to slice it differently and have it, or they used to add it for a cooking along with vegetables to have flavors. So this is a kind of a disease. And so then we have this disaccharides, sucrose, lactose, and you can't say it as a disease, but it is also a habit, it could say, habit of certain peoples. And we have the hedgehog, and the flower of touch me not plant, and pin cushions with pins. So beautiful flower was it. And the lactose. And so they have not given the picture. And we have the isomaltase and polysaccharides, and we have the plant kingdom, the structure of starch. Oh my god, I did not learn this, but I mentioned starch 1,000 times in my biology paper. Vasudevan would be really unhappy with me. And so usually among the hundreds, I am a girl also, and at the same time I am from a military batch. So they would have certainly expected me to ask questions and put a case on starch. But I usually see the plants, usually was along with the curar plants. So I usually break those plants and see the starch. So I write about the starch. And but anyways, so we glycogen, cellulose, and inulin and dextrans, and plasma volume expanders and the chitin, and exoskeletons of crustaceans and insects, this chitin. And we have this heteroglycans, the agar, agarose. It's certain we must learn a few of these words to be familiar with the media, and also the geographical and animals and their structural proteins, and also this mucopolysaccharides here, glycosaminoglycans, and the heteropolysaccharides. I've read 1,000 times on these names. And we have the hyaluronic acid, and it's present in the connective tissue, tendons, synovial fluid, and vitreous humor, and a lubricant in the joint cavities. And actually, in the joint cavities, is that acid? No, it's not acid actually. Why he has written hyaluronic acid? Maybe somewhat like acid. We'll see. That is N-acetyl glucosamine, beta 1,4-glucuronic acid. So it is a glucuronic acid. So it's glucose is usually a base, and N-acetyl glucosamine, maybe an acid base with a few of acid qualities. So joint cavities, and so if it is an acid in the joint cavity, the joint would burn, it would not remain stable. And so then we have the heparin and anticoagulant, and so idose is the fifth epimer of glucose. So we have the chondroitin sulfate, so the ground substance of connective tissue, distributed in the cartilage, bone, tendons, and the cornea and the skin. So the sulfate crystals, usually these are black. Sulfate is usually black, and chondroitin sulfate, and chondroitin is about chondro means something like cartilage, chondrite cells, like, and so the composition is repeating units of glucuronic acid. It's beta 1,3 N-acetyl, galactosamine sulfate, beta 1,4, and so on. So this N-acetyl, acetyl is CH3, right? 3N acetyl galactosamine. So CH3CO, right? CH3CO is acetyl, and carboxyl group. And I love these constellations and planes, and I enjoy writing these, and have made and have given some of the new structures also in my 12th standard chemistry. So we have given written formulas for new structures also, and names have still to be given, and the author already had special left names also, and I know he will make it next 2 years. I will see in my sister's book also, it will be available in the Tamil Nadu syllabus. Now the 10th standard is available. 10th standard edition, what we all have written, have been available. I think next 12th standard it will be available. So I just went into the history and geography of the 10th standard, the first volume, and it was okay. We are done. And so then we have the keratin sulfate. does not contain any uronic acid and galactose and unacetyl glucosamine and beta linkage. It's found in cornea and tendons. Um, um, just because I I have not been introduced these into as somewhat to inherit the author's words. I have to take this I was given the what do you say this accountant work. So actually my mother's father would have been an accountant, so I inherit his job. So they show me accounts of all of these, but they never knew that I learn all of these. But it's certain we could not remember it for lifelong or for after a period of 2 to 3 years. I'm just remembering it just because I saw the book. So it doesn't matter. And then we have this L-iduronic acid and N-acetyl galactosamine in beta 1,3 linkages, skin, blood vessels, heart valves. And so if you see a book, a 12th standard book, it will certainly look it's a short book and you could do everything with it. But if you go deep into chapters, you'll just will have time only for some first 10 chapters. Then again you'll go insane. So next we have chapter 8, chemistry of lipids. So the classification of lipids, and we have simple, compound, derived, lipid complex and fatty acids, aliphatic, carboxyl acids. And so one second, I'll just so keratin sulfate is found in the cornea and the eyes. This dermatin sulfate is like skin, right? But sulfur, I need to go again this logbook. So if you are very much interested, take the logbook, go into the periodic table, and you have a app in the cell phone the periodic table. So you see the structures and molecules, and get into memorize all of that with the pictures. So if it is along with the pictures and the structure, you could certainly memorize everything, and it will be very easier to deal with the all of the mining and mining work. The field especially concerned about the mining, the land and the stones and its form, and the molecules and the molecular structure and the function and the uses, and all of the Wikipedia items. So next we have this chemistry of lipids, chapter 8. So then we have chapter 9 and all of that. Let me see how much chapters we have. We're having totally 50 chapters. So I'll move up to some 10 chapters today. And so classification of lipids. So we have the palmitic and the stearic acid. The author is again concerned about the ration, the government ration, the acetic acid, butyric acid, palmitic and stearic acid. So we have all of these industries along in the Trichy main road. So then we have the unsaturated fatty acids and the cis configuration, and linolenic acids, prostaglandins. 1,000 times on the road, I have never met all of these industries, and taken photos and had a conversation with them. It's very sad, but previous batch when we were there on action, we had lot of these interactions and all. But simply just we can't go and have their identities, isn't it? So they somewhat they might be scared or not willing to tell, or like they might have a special code and formula. And so the properties of fatty acids are like hydrogenation, halogenation, melting point, salt formation, and triacylglycerol, and ester formation and oxidation of fatty acids. The neutral fats, we have carbon atoms with the triglycerides and triacylglycerols, and we have adipose tissue storage and hydrolysis and saponification and iodine number and all of that. And we have rancidity of fat, hydrolytic rancidity and oxidative rancidity. So they have a tendency to become rancid, appearance of unpleasant smell and taste for fats and oil, just because they get oxidized. So immediately after extraction, when exposed to air, they become unpleasant, and they have life. So hydrolytic rancidity is due to partial hydrolysis of triacylglycerol molecules due to traces of hydrolytic enzymes present in naturally occurring fats and oils. So traces, because of these traces. And oxidative is due to partial oxidation of unsaturated fatty acid with the resultant formation of epoxides and peroxides of small molecular weight fatty acid by peroxides and free radicals. So there are formation of epoxides and peroxides. Epoxide is single oxide, peroxide is double oxide. So if it occurs in vivo, it will affect integrity of biomembranes, leading to cell death. And so then we have this repeated heating of oils, formation of polymerization of cyclic hydrocarbons, and then we have axis, then we have phospholipids, then we have phosphatidates, and amphipathic nature and micellar formation and liposomes, biomembranes. Then we have phosphatidylcholine or lecithin, and PUFA, and lysolecithin, and ethanolamine cephalin, phosphatidylinositol, and plasmalogens, and glycerol, and sphingolipids, and phospholipids. The sphingolipids, sphingo means spinning or round halo, and have a long aliphatic amino alcohol sphingosine, and it's attached to a fatty acid and amide linkage to form a ceramide. So, and so the chain length is 18 to 24. So then all of these things, and glycolipids, gangliosides, sulfolipids, sulfatides. So the ceramide, I think he is mentioning about the plaque. gastrointestinal, and the organic outside skin plaque, lipid storage diseases. And so then we have section B, general metabolism. Okay, thank you. Have a nice time.